Welcome to this explainer on acute lymphoblastic leukemia, or ALL. We're diving straight into what is, honestly, a massive medical paradigm shift. We're tracking this wild journey from an era completely dominated by highly toxic, heavy-handed chemotherapy to a brand new age of elegant, targeted treatments. It's fundamentally changing what it means to get this diagnosis.
To really grasp where we are right now, we gotta ask a question that's puzzled oncologists for decades. Why do children survive leukemia at much higher rates than adults? I mean, historically, childhood ALL cure rates skyrocketed. We're talking up to 80 or 90%, while adult survival just lagged way behind. Well, today we're uncovering exactly why that happened, and more importantly, how modern science is rapidly closing that gap. Okay, here's our roadmap for today's explainer. First, understanding ALL today. Then, the genetic fingerprint, followed by tracking hidden leukemia, or MRD. After that, we'll look at the cost of chemotherapy, the immunotherapy revolution, and finally, wrap up with a new era of survival. Let's get to it.
Starting off with Section 1, Understanding ALL Today, and Diagnosing a Rare Malignancy.
When diagnosing ALL, what we're actually looking for are lymphoblasts, essentially taking over a diagnostic sample. ALL is a rare clonal lymphoid malignancy. Basically, these abnormal, immature white blood cells just multiply like crazy, totally crowding out the healthy cells in the bone marrow and blood. And while about half of newly diagnosed ALL patients are kids, this disease absolutely strikes across the entire age spectrum, right into older adulthood.
But confirming this diagnosis, it isn't as simple as just peering through a microscope. It actually requires a really strict diagnostic triad. First up, assessing the physical morphology, so what the cells literally look like. Second, we have to identify the immunophenotype using flow cytometry. That tells us that the leukemia originated from B cells or T cells. And third, and this is absolutely crucial, we need to uncover the specific cytogenetic and molecular profile. The DNA errors hidden inside these cells, they hold the key to everything else we're going to talk about.
Moving on to Section 2, the genetic fingerprint, and identifying the true villains.
Now, there is a fascinating story behind the Philadelphia chromosome. This is a very specific genetic rearrangement, basically a fusion between two genes called BCR and ABL1. Now, historically, if a patient had Ph-positive ALL, it was practically a guarantee of a terrible prognosis. It was literally the most unfavorable subtype you could possibly get. But science completely flipped the script here. Because we identified this exact genetic fusion, researchers were able to create targeted tyrosine kinase inhibitors, or TKIs. Think drugs like imatinib, dasatinib, and ponatinib. These drugs act like a homing missile for that exact fusion, turning a once deadly marker into a highly manageable condition.
So here's the catch. While we've largely tamed the Philadelphia chromosome, we also have to be on high alert for something called Phi-like ALL. This one is incredibly tricky. Its gene expression profile mimics Phi-positive ALL perfectly, but it totally lacks that actual BCR-ABL1 mutation. Because of this clever disguise, it's historically been highly resistant to standard chemotherapy. We see it a lot in adolescents and young adults, and it means we have to dig even deeper into the genetics to find alterations in other pathways, like JAK-STAT or ABL class fusions, just so we can figure out how to target those instead.
Which brings us to Section 3, Tracking Hidden Leukemia, also known as MRD, or Measuring Residual Disease. You see, even when a patient looks to be in complete morphologic remission, meaning we can't find a trace of cancer under a standard microscope, hidden leukemia can still linger. And if a patient hits remission but still has this MRD, their chance of relapse goes through the roof. To track this, we rely on some incredibly sophisticated tools. Flow cytometry is fast and broadly available, capable of finding one single leukemia cell in 10,000. Quantitative PCR pushes that to 1 in 100,000. But next-generation sequencing, or NGS, that is the ultimate sniper. It can sequence B or T cell receptors to find just one single leukemic cell hiding among a million healthy ones. It's wild. We use these ultra-precise tools to track the disease throughout the entire treatment journey.
All right, Section 4, the cost of chemotherapy and its toxic legacy. For decades, the only way we knew how to clear this microscopic residual disease was through sheer medical brute force. I mean, traditional regimens were basically carpet bombing the body. Patients had to endure 15 or more incredibly harsh chemotherapy agents spread across intense induction, consolidation, and maintenance phases that could last up to 3 years. The toll was just massive.
We're talking severe myelosuppression, which is a dangerous crash in bone marrow activity, huge risks of fatal infections, long-term organ damage like cardiomyopathy, not to mention an immense socioeconomic burden. But today, we can directly contrast that toxic legacy with the elegance of novel targeted agents. These new drugs attack specific leukemia markers while drastically, and I mean drastically, reducing systemic toxicity.
And historical data really lays bare the true cost of that traditional chemotherapy, especially for adults. Remember earlier when we asked why kids survive more often? Well, it's largely because children could physically tolerate those massive cumulative doses of steroids and myelosuppressive drugs. Adults and older patients, they simply couldn't handle that level of toxicity. Their bodies just couldn't take it, which led to significantly lower historical survival rates. We desperately needed a smarter approach.
That leads perfectly into Section 5, the immunotherapy revolution and training the immune system.
Think about it. What if we could bypass that extreme toxicity completely? What if, instead of poisoning the whole body to kill the cancer, we could just train the body's own immune system to do the heavy lifting? Well, welcome to the immunotherapy revolution. We've got a totally new arsenal now. Take blinatumomab, for instance. It's a bispecific T-cell engager. It literally acts as a bridge. It grabs a healthy immune T-cell with one arm, and it grabs the CD19 protein on a leukemia cell with the other, essentially forcing your own immune system to destroy the cancer. Then there's inotuzumab, which acts like a microscopic Trojan horse, binding to the CD22 marker on leukemia cells and dropping a toxic payload directly inside. And of course, the famous CAR T-cell therapies, where we extract a patient's own immune cells, engineer them in a lab to become specialized leukemia hunters, and infuse them right back in.
So how does all of this play out in actual clinical practice? Well, when you combine a targeted TKI, specifically ponatinib, with the immunotherapy blinatumomab for newly diagnosed Philadelphia chromosome-positive ALL, the results are massive. By course 3, patients are seeing an absolutely astounding 90% complete molecular response rate. Let that sink in. In 90% of these patients, the leukemia becomes completely undetectable. And the absolute best part, they achieve this without relying on heavy, intensive chemotherapy.
Finally, Section 6, a new era of survival with skyrocketing success rates.
The clinical data we're seeing right now is just the ultimate payoff. Today, 5-year overall survival is hitting 75% for adult patients treated with a combo of standard intensive chemotherapy, known as hyper-CVAD, and that targeted drug ponatinib. I mean, just a couple of decades ago, an adult diagnosed with Philadelphia chromosome-positive ALL faced a highly fatal disease. It was grim. But now, thanks to the deep molecular remissions brought on by adding these targeted TKIs, 3/4 of these patients are surviving long-term. It's a total game changer.
And amazingly, the news gets even better for older patients. Take the D-ALBA study, which combined the TKI dasatinib with the immunotherapy blinatumomab. It shows that even older adults, the exact patients who previously could not handle traditional intensive chemo, are now seeing long-term overall survival rates near 80%. They're hitting these incredible numbers by using smarter, chemotherapy-light approaches that spare their organs and actually preserve their quality of life.
Which leaves us with this incredible, provocative thought to close on. We've seen how tracking the disease down to a single cell and unleashing the immune system has fundamentally rewritten the rules for acute lymphoblastic leukemia. As precision immunotherapies and targeted agents continue to knock traditional, heavy chemotherapy out of the picture, are we truly standing on the threshold of an era where acute leukemia is routinely cured without the devastating toxic cost? The data, quite frankly, suggests we are well on our way. Thanks for hanging out with me for this explainer, and keep questioning what's possible in the future of medicine.